Welcome to our Bioprocess Engineering online tutorial on how to perform a bioprocess lifecycle assessment on spreadsheets. Through the Bioprocess Engineering online tutorials, you learn the principles of bioprocess engineering and their applications to enzymatic bioconversions, microbial fermentations, and cell cultures bioprocesses. For more details, visit us at bioprocessdigital.com. You also learn how to use spreadsheet software for key bioprocess engineering tasks, including analyzing experimental results, modeling and simulating bioreactors, designing and optimizing bioprocesses, evaluating economic and environmental performance. In this tutorial, you'll learn why to conduct a bioprocess lifecycle assessment on spreadsheets and how to construct a LCA spreadsheet simulator for a bioprocess. The spreadsheet construction is illustrated on the example of the glutamic acid fermentation process. Life cycle assessment, LCA, is the prevailing methodology employed to evaluate the environmental footprint of enzymatic or cellular bioprocesses. LCA examines the environmental impacts associated with the materials and energy resources used, as well as the wastes or emissions produced. For bioprocesses, LCA typically evaluates three key environmental impacts: primary energy consumption (PEC), measured in megajoules; water consumption (Watt), measured in kilograms of water; global warming potential (GWP), measured in kilograms of CO2. Bioprocesses display a wide range of impact indicator values. Per kilogram of generated product, the primary energy consumption indicator typically ranges from 10 to 10,000 megajoules. The water consumption indicator ranges from 1 to 10,000 kilograms of water. And the global warming potential indicator varies from 1 to 1,000 kilograms of CO2. Indeed, these environmental impact indicators are influenced by various factors, including bioprocess flow sheet or sequence of unit operations, the industrial equipment utilized, raw materials, chemicals, and water used, energy sources such as electricity, gas, or fuel, equipment operation modes such as batch or continuous, bioprocess operational variables like concentrations, flow rates, and temperature, equipment cleaning and sterilization procedures. To address the multitude of factors impacting the environmental footprint of a bioprocess, the bioprocess engineer develops LCA simulation models capable of predicting the environmental impact indicators for specified bioprocess flow sheet and operational conditions. These predictive models serve as valuable tools for optimizing bioprocess operations to minimize environmental impact indicators, guiding the pursuit of the lowest possible environmental footprint. Bioprocess lifecycle assessment simulation models are optimally crafted through spreadsheet software. Typically, the LCA simulator comprises 4 interconnected sheets: bioprocess operation, material inventory, energy inventory, and impact indicators. Within the spreadsheet, the bioprocess operation conditions, outlined in the first sheet, are leveraged to compute both the bioprocess materials and energy inventories. These inventory results are then utilized to calculate the environmental impact indicators generated by the bioprocess. The first spreadsheet in the Bioprocess LCA simulator is the Bioprocess Operation Sheet. This sheet contains all pertinent data concerning the bioprocess flow sheet and operations, which directly impact the material and energy inventories throughout the process. The Bioprocess Operation Sheet outlines the bioprocess flow sheet, that is the sequence of upstream and downstream operations that transform raw materials into the desired product. For instance, in the glutamic acid fermentation process, the bioprocess includes the operations of media preparation, sterilization, fermentation, centrifugation of cellular biomass, concentration of the glutamate solution via evaporation, crystallization of glutamate, and drying of the final crystals. In addition, the Bioprocess LCA also considers the equipment cleaning and sterilization. For each unit operation, the industrial equipment utilized has to be specified. For the glutamic acid production process case study, the central equipment is a 200 cubic meter fermenter. Supplementary equipment comprises a plate heat exchanger utilized for sterilization, a disc centrifuge, a film falling multiple effect evaporator, a crystallizer, and a rotative dryer. On the bioprocess spreadsheet, each unit operation under consideration is accompanied by detailed specifications outlining the operational conditions that impact both material and energy inventories. These conditions are first the operational mode of the equipment, distinguishing between batch and continuous processes. Furthermore, these specifications encompass a range of operational variables, including medium temperature, operation duration, component concentrations, and medium flow rate in the case of continuous operations. For example, in the case of batch medium sterilization, the operational variables specify the precise temperature required for sterilization, alongside the duration of the operation. In bioprocesses, the biotransformation step stands out as the most sensitive unit operation to operational variables. The final performance of enzymatic or cellular transformations are strongly dependent on both the kinetic characteristics of the employed enzyme or cell and on the operational conditions of the bioreactor. Within the bioprocess flow sheet, it is imperative to provide data concerning the stoichiometry and rates of the biotransformation reaction. For instance, for the glutamate fermentation, crucial information includes the conversion stoichiometry coefficients of glucose, ammonia, and minerals into biomass and glutamate, along with the maximal specific rates of cell growth and glutamate excretion. In enzymatic or cellular biotransformations, additional bioreactor operational variables influence the operation of the process, notably temperature, substrates concentrations, and process time. For example, for the batch glutamate fermentation, it is necessary to specify the initial sugar concentration in the thermal induction time for glutamate excretion, as well as the fermenter agitation power and aeration rate. Furthermore, the spreadsheet must specify or calculate the outcomes of biotransformation under fixed operational conditions. This includes determining the fermentation time necessary for sugar utilization and the resulting final concentrations of biomass and metabolite. The second spreadsheet in the LCA simulator is the material inventory sheet. This sheet assesses the quantities of materials utilized throughout the various bioprocess unit operations. Based on the process parameters outlined in the preceding bioprocess operation sheet, the material inventory calculates the amounts of raw materials, chemical reactants, and water consumed, the quantities of products and byproducts generated, the quantities of liquid effluents, solid wastes, and air emissions produced. The material inventory assesses the material balance for each bioprocess unit operation. This entails calculating the quantities of materials entering from the preceding operation and exiting for subsequent ones. Additionally, it evaluates the material inputs, such as raw materials or reactants, and outputs, including wastes and emissions, for each unit operation. In the case study of the glutamic acid process, for example, for the initial media preparation operation, the spreadsheet assesses the required input quantities of sugar, ammonia, minerals, and water. During the fermentation operation, the inventory examines the volumes of air entering and exiting the fermenter, along with the amounts of consumed oxygen and emitted CO2. Through the aggregation of mass contributions from the involved unit operations, the mass inventory spreadsheet establishes the comprehensive material inputs and outputs for the entire bioprocess. This inventory is conducted initially per batch operation and then per kilogram of generated product. For instance, In the case of the glutamate process, a single batch operation yields approximately 20,000 kilograms of glutamate. For every kilogram of glutamate produced, the process necessitates 1.8 kilograms of glucose, 0.15 kilograms of ammonia, 0.03 kilograms of minerals, and 12 kilograms of water. Simultaneously, 0.9 kilograms of CO2 and 3.1 kilograms of liquid effluents are generated. The third spreadsheet in the LCA simulator is the energy inventory sheet. This sheet assesses the quantities of energy utilized throughout the various bioprocess unit operations. Utilizing the process parameters, detailed in the bioprocess operation sheet, and the processed masses evaluated in the previous material sheet, the energy inventory calculates the energy consumption for each unit operation equipment. These operations consume mechanical and thermal energies, which can be supplied via electricity, gas, or fuel sources. The spreadsheet assesses the mechanical energy required for tasks such as tank agitation or fluid pumping within equipment. For example, in the case of fermenter agitation, the necessary mechanical energy, measured in megajoules, is determined by multiplying the volumetric agitation power by the medium volume and the agitation time. Subsequently, the consumed electrical energy, measured in kilowatt hours, is computed by dividing the mechanical energy by the motor's electricity to mechanical conversion efficiency and by 3.6 to convert units appropriately. In thermal unit operations like sterilization, evaporation, and drying, the energy spreadsheet additionally calculates the thermal energy needed for fluid heating or water evaporation. For medium sterilization, for instance, the required heat is determined by multiplying the medium mass by the medium heat capacity and the temperature rise. When heat is supplied through an exchanger by condensing steam produced in a gas boiler, the necessary gas volume, measured in cubic meters, is calculated by dividing the heating energy by the boiler efficiency and the gas burning heat per cubic meter. By aggregating the mechanical and thermal energy consumptions across all involved unit operations, the energy inventory spreadsheet determines the comprehensive energy usage for the entire bioprocess. Initially, this inventory is conducted on a per-batch basis and subsequently normalized per kilogram of generated product. For example, in the glutamate production process, each kilogram of glutamate produced necessitates 1.6 kilowatt hours of electricity and 0.35 cubic meters of gas. Furthermore, when electricity and gas consumptions are converted into megajoule energy equivalents, the spreadsheet also offers a ranking of unit operations based on their energy consumption. In the case of the glutamate process, evaporation, sterilization, and fermentation emerge as the most energy-intensive operations. The fourth spreadsheet in the LCA simulator is dedicated to the impacts indicators analysis. This sheet evaluates the impact indicators resulting from the entire bioprocess and its constituent unit operations. Based on the results from the preceding material and energy inventories, the impacts indicators spreadsheet computes the three indicators: IPEC for primary energy consumption, IWatt for water consumption, and IGWP for global warming potential. These calculations are conducted for both the entire bioprocess and on a per kilogram basis of product. The impact indicators spreadsheet initially specifies the impact factors associated with the material and energy inputs and outputs, analyzed in the material and energy inventories. These impact factors quantify the environmental impacts generated by the production, transportation, and utilization of one unit of material or energy sourced from their respective life cycle assessments. For example, per kilogram of glucose, the impact factors are approximately 25 megajoules for peck, 6 kilograms water for watt, and 1 kilogram carbon dioxide for GWP. And per kilowatt-hour electricity, the average European impact factors are 7 megajoules for peck, 4 kilograms water for watt, and 0.23 kilograms CO2 for GWP. The spreadsheet computes the impact contributions of materials and energy inputs and outputs for each of the three considered impacts. This contribution for each component is determined by multiplying its quantity MI by its respective impact factor FI. The overall impact indicator of the bioprocess is then obtained by summing up all these individual contributions. Following this, indicators per kilogram of product are calculated by dividing the total impact indicator by the mass of the produced product. Additionally, the spreadsheet assesses the percentage contribution of each material and energy component, offering a ranking based on their contributions. In the case of the glutamate process, the spreadsheet indicates the following indicators per kilogram glutamate for the three assessed impacts: 75 megajoules for the primary energy consumption. The two most important contributions are from glucose and gas, 30 kilograms water for the water consumption. Main contributions are from fermentation media water and glucose, 4.5 kilograms CO2 for global warming potential. Most important contributions are from gas, glucose, and fermentation emitted carbon dioxide. Accordingly, for the three impacts indicators, it is the production of the glucose used in the fermentation medium, the water for media preparation, and the gas for fluids heating that bring a dominant contribution to the glutamate process environmental footprint. Furthermore, when evaluating the individual contributions of unit operations to material and energy inventories, the spreadsheet analysis can extend to include an assessment of their relative impacts on the three indicators. In the context of the glutamate process, the foremost contributions to PEC, watt, and GWP stem from the initial media preparation operation, primarily attributed to the consumption of water and media components. Significant contributions also arise from operations such as evaporation and sterilization, owing to their substantial thermal energy consumptions. Additionally, fermentation plays a notable role due to its energy-intensive agitation and associated CO2 emissions. In summary, throughout this tutorial on life cycle assessment of bioprocesses, you learned bioprocess LCAs are effectively conducted using a suite of interconnected spreadsheets tailored specifically for bioprocess operations, material and energy inventories, and impact indicators. The LCA spreadsheets facilitate a comprehensive simulation of how operational conditions within bioprocesses influence environmental impact indicators. Additionally, it enables a detailed analysis of the contributions of the unit operations material and energy resources to these impacts. If interested, you can gain a deeper insight in the LCA methodology by enrolling to one of our online courses: Introduction to Bioprocess Lifecycle Assessment and Lifecycle Assessment of Bioprocesses and Bioproducts. For further information, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com. We hope you found value in this introductory tutorial on bioprocesses. Your feedback is important to us, so please share your comments with us. Explore our website for more information about our comprehensive online courses in bioprocess engineering, and be sure to subscribe to our Bioprocess Engineering online channel for upcoming tutorials. Thank you for watching.